originally developed as antiretrovirals, and reduce liver damage. For HSV, the virus can be suppressed with drugs like acyclovir. But for all of these, once therapy stops, the virus tends to come back. That's almost inevitable for HIV. In about 2 weeks, virus will come back. Hepatitis B will start back up again. And when people go off of antivirals, um, recurrences tend to go back to their kind of original frequency. So we'd love to have some way of curing these viruses rather than just controlling them. Sort of the metaphor I use is if you've got weeds in your garden, if you're just plucking the tops, your garden might look fine, but it's a lot of work and you can sort of never stop. So this is the way to really get down into the roots of these infections and dig them out. And the metaphor is there, but what this would really mean in terms of the virus is going after these long-lived DNA forms. You know that HIV integrates into the chromosome of host cells. Hepatitis B has this covalently closed circular form that lives in hepatocytes. And for herpes, the virus establishes latency in neurons that innervate the region that was originally infected and just lives there as episomes for the life of those cells. And you probably know that your neurons persist for life, so therefore the virus has found this perfect place. So if you think about cures for these infections, you could try to eliminate the infected cells. Probably not a great idea for your neurons because you don't make very many new ones. So is there a way to actually eliminate the infectious virus from the cell? Because if you could do this, there would be tremendous clinical benefit. So we've been using gene editing as an approach for this. I think everybody knows about gene editing, and people generally know about CRISPR-Cas over here on the right. A point that I always try to make in the meeting is that CRISPR-Cas is not the same thing as gene editing. It is a tool for gene editing, and there are other molecular classes that can specifically recognize DNA and cleave it at specific sites. And people probably know about zinc finger nucleases. There are tau effector nucleases that had a brief moment in the sun. We've worked a lot with meganucleases because these are really highly specific and they're very, very small. So these pictures are roughly to scale. I should probably do it at some point so they actually are truly to scale. But these are, despite the name mega, are actually tiny little proteins, which makes them very easy to vector, to put them into AAV vectors or whatever else you want to do. They're very difficult to retarget for a desired specificity. So we know how to do that. And it usually takes a couple of months if we have a new target. I mean, as you probably know, if you have CRISPR-Cas and you want to target something new, you can kind of be doing it tomorrow once you just order the single guide RNA. For meganuclease, it's a bigger process, but once you have it, you have this really ideal reagent. So we've been working on those. And sort of the general idea is that you have a virus, and we have a meganuclease that can cut it. And if the virus were an episome, you would get a cut, and then that would be repaired by the cell. And through repeated rounds of repair and cutting, at some point you get an indel there. And if that were in something that was important to the virus, you would be knocking out the ability of the virus to replicate. And I'll show you some data where we relied on that in the early days. What we subsequently discovered is that if you cut an episomal virus twice, the cell has a really hard time kind of grabbing these two pieces and putting them together properly. And what typically happens is DNA degrades. So a lot of times what you'll see is that we're going after a virus. We just want to not only inactivate it, but ideally just completely get rid of it. And so we're typically going to try to cut it in two places. So HSV latency, let's just talk a little bit to bring everybody up to speed on HSV and how it works. So the virus infects at a mucosal or skin surface, and it gets access to the nerves that innervate that region. And the virus moves down the axons, it jumps onto the molecular motors that move things up and down the axons. And it accesses the nerve cell body, which is in a ganglion. Here it's a trigeminal ganglion, but it could be a dorsal root ganglia along the spinal cord. And then it establishes these latent episomes. Later on, those can reactivate, and viral components will go back down the axon, be assembled into virions, and be released into the mucosal surface, where they then can replicate to high levels. They can shed, they can attempt to infect a new host, they can cause a symptomatic lesion. So this is where all the disease is happening. But the really the long-lived forms are way down here inside the body. So a ganglion has about, we'll say, 10,000 neurons. So it's actually not many cells. You think about HIV as infecting potentially millions, hundreds of millions of cells. There's relatively few neurons here. Only about 10% or so are infected typically. And then an infected neuron might have 10 or so copies. So there's not very much HSV, and I'll actually show you this in a second. Um, but this reactivation from ganglia is what we'd like to stop. And that process is largely influenced, heavily influenced by just the amount of latent virus here. So unlike a disease process like HIV, where you sort of think about having to get rid of every single virus, otherwise you kind of have gotten nowhere. Here, if you can just reduce that viral load, you in theory could have a tremendous effect on reactivation. So there's a lot of things about herpes, sort of this anatomic localization of the reservoir, the small size of the reservoir, and the fact that the reservoir doesn't have to be completely reduced for tremendous potential benefit makes this a really nice target for gene editing as an approach to durable control or even cure. So I mentioned that it's anatomically localized. This is a trigeminal ganglia. Dan Strongen in the lab, shown here, has developed some optical clearing approaches. So you can actually take this ganglia out of an infected mouse. Then you optically clear it, and it ends up looking like this, which you can hardly see in this picture. And then you can just take a series of DNA probes, Z probes, that then amplify the signal through these sort of complex dendritic structures with a bunch of fluorophores on them. But it just illustrates the point. I actually have a movie for this, but I never show it in talks because it will always crash my talk, it seems. But you can sort of see just some screenshots. All these dots, this is the reservoir. This is sort of all the disease is caused by these limited number of infected neurons within this. So it is a small reservoir. It is in a place we know how to get gene therapy, gene editing. So it really does seem like a wonderful target. We use HSV1 specific meganucleases. The ones that I'll show you became available to us through a collaboration with a French company called Selectus. And we have mainly used three of them. One called M5, stands for meganuclease 5, M8, and then M4. And you can see what they target here, sequence within the major capsid protein, the viral polymerase, or M4 that I'll show you a fair bit of data on, targets a duplicated region within the locus. This appears twice in the genome. And so if you sort of think about that, we'll use these in combination to get those two cuts I mentioned. But for M4, one enzyme can give you those two cuts. You sort of get more bang for your buck with that. We have some controls as well. And I mentioned how these things work. So this has been, for better or for worse, a decade plus long process now, sort of turning this into something that's real. And we just started with evaluating whether these meganucleases actually could cut latent HSV. They weren't really originally designed to target HIV. It was a little bit of a serendipitous observation that they did. That's where we got involved. Um, and basically, we established a culture model that was fairly complex based on just adherent cells, and you can infect them in the presence of interferon and acyclovir and do some heat shock and so forth. Then you could maintain latency over an extended period. But then we can introduce a meganuclease into this where there's no active viral replication, but the virus is latent, and just say, can we get any gene editing? We'll pull the virus out. We will look then by sequencing, and the answer was yes, you could gene edit this. It wasn't very efficient. We edited less than 10% of the latent virus in there. Again, this is kind of depending on these indel formation with a single nuclease, but it did show that they worked, and that they did work on the latent form of the virus, because that's what we'd really like to target. So we spent a fair bit of time thinking about how we could actually translate this into something that might work in a living organism. And we knew how to infect mice with HSV. So we asked, could you actually get something to the proper neurons using an adeno-associated virus vector? And some of our early work was using AAV1. So remember, AAV comes in various serotypes, typically just has AAV and then a number or a combination of letters here. And so if you just look at the trigeminal ganglia of these mice. You pull them out, the new end simply targets the neurons. So you can sort of see a lot of the trigeminal ganglia is actually just axonal tracts that don't really stain here, but you can see the nerve bodies. And then if we just have AAV deliver a marker, in this case, M-cherry, that we're detecting via immunostaining, you can see nice production here. So we can get things in a mouse to the right place, and we use that approach then to really see whether we could attack a latent HSV infection in vivo. And so the basic model that I'm going to show you probably the next 10 slides, permutations of this, is that we infect mice through various routes. A very common route is through the eye. We give them the proper dose of HSV. Then we wait for about a month, and this lets the acute infection settle down, and the mice establish a latent infection. It genetically looks just like latency in humans, that there's only one herpes gene transcribed, something called LAT, the latency associated transcript. Otherwise, the virus is completely latent, quiescent. And I should say that in mice, that virus doesn't reactivate. So it just sits there. It won't come back out like it would in a human, but it's there. And so you can try to target it. So we introduced AAV1, like I just showed you, put it into the whisker pad of mice. Those neurons go to the same neurons that the eye, that come from the eye. We'll wait a month to let gene editing happen, and we'll take the ganglion out, deep sequence or sequence the virus, and just ask, did we get gene editing in vivo? And the answer here again was yes, we can gene edit latent virus in vivo. But again, it was pretty low efficiency. So here, kind of one of the better animals was 4%. So 2 to 4%, not great, but I still think, as far as I can tell, this was the first demonstration of gene editing of a true latent infection in an animal model. So it was certainly advanced, but clearly not something that was going to, editing 4% of somebody's virus is probably not going to dent disease. So sort of went off and spent the next couple of years really trying to optimize the entire process. Things like looking at different AAV serotypes, looking at different doses, looking at different routes of inoculation, sort of all these things. And one thing that we came to realize is you can also put the AAV in, you can put it through the eye, you can put it retroorbitally, so actually in the bloodstream. There's a variety of approaches. And also not all AAVs are created equal. So by single cell analysis, different AAVs of these types actually go to different neurons. And so we tried combinations of AAVs and combinations of multiple meganucleases so that we get those two cuts I talked about. And we ultimately found here in this experiment that if you used combinations of different AAVs to try to cover all the neurons, and you made these double cuts using two meganucleases, you could not only gene edit HSV, but it actually began to disappear. And so if you looked at the viral load in treated animals, so here's the control animals, it was reduced by about a log, 90% in the ganglia of these animals. This is the superior cervical ganglion. This is one of the important ganglia that's infected during latency. We've always seen that SCG gene edits better than TG. That happens in every experiment. See, the percentages here are lower. Typically, we'll get about 60% reduction here. But you can see this, that now you can start to achieve not just gene editing, but elimination of viral genomes from latency. They're just gone at levels that you might start to think could have some sort of clinical or therapeutic benefit. So I mentioned that HSV infects mucosal surfaces, so there's orofacial herpes typically caused by HSV1. There are genital infections that can be either HSV1 or 2. And then I showed you sort of this difference between the superior cervical ganglia tend to be edited better than the TG. That really seems to be just due to access of AAV to those ganglia. But we wondered in genital infections, which does it look like? Is it pretty good like SCG, or is it a little less so like TG? So we did two experiments just side by side. We did intravaginal infections of mice with HSV, or again in parallel, just these eye infections. So maybe start with the eye. Here's the reduction. So we have this 90% reduction in the SCG, and the TG, we have this 61% reduction, just like we almost always see. I'll mention that of the virus that stays after this, a substantial fraction of it has actually been mutated. So it's actually knocked out, even though it's still there. So you kind of and I think that happens because if the two cuts are sort of out of sync and there's kind of one happens, if the cell can repair that before the second cut happens, you can have mutagenesis there. So that's what happens. But if you look in the genital tract of the mice, it turns out that the DRG are reproducibly actually the very best ganglia and are extremely accessible. And we typically get this 97, 98, 99% reduction in the viral load there. So good news if we're thinking about ultimately a therapy for genital herpes is that it's actually even easier than our original results in oral facial infection. So people were pretty excited about this. You're starting to get rid of 98, 99% of the virus. But of course, for a person living with an HSV infection, simply reducing their ganglionic load doesn't necessarily reflect clinical benefit. And I mentioned that mice don't reactivate HSV. So you take a mouse and you just, in this case, inject it with PBS. There's no viral shedding because mice don't reactivate virus. But if we inject virus with a bromodomain inhibitor, JQ1, we do this intraperitoneally. This came to our attention through HIV studies where it was used as a latency reversing agent. This compound actually induces reactivation of herpes, and that reactivation can be detected simply by swabbing the eyes or the genital tract of the animals and then doing PCR on the swabs. And so about 40 to 50% of animals getting this dose at any given dose will reactivate. You can redo it and sort of it'll be a separate subset of the animals. It's kind of very stochastic, just like human recurrences. But this gives you a model system in which one can hope or contemplate looking at the effect of viral load reductions on shedding. And shedding is something people care about because that is correlates well with lesions, and it also, of course, correlates with the risk of transmission. And the answer to does this matter is yes. So if we take mice, we infect them with HSV1 just like we've always done. We give them our meganuclease therapy. Wait, in this case, we wait a month and a half. We do the JQ1 reactivations. Well, first of all, if you just look after all this is over at the ganglia of the mice, we have these reductions just like we always see the SCG, 90-ish%. Maybe even a little less in this experiment. It's 50 to 60% of the TG. But this actually has a tremendous effect on the ability of the virus to reactivate. So here's our control animals. There's about 12, 14 in the group. You see that about half reactivate after each of the two, more after the first one than after the second, but only a single animal out of the treated groups actually reactivated. So not only do you reduce the viral load, but you reduce the likelihood of reactivation. Now, we've gone on to really simplify things. I mentioned there were 3 different AAVs. In each AAV serotype, half of it expressed one nuclease, M5, the other half expressed M8. So there's 6 things all going in at once, very complicated. We have actually simplified this down subsequently that M4, which achieves this double cut with just one nuclease. And we also identified an AAV serotype called AAV9 that's actually extremely good at getting to ganglia in mice. It turns out also in humans, it's the basis for approved gene therapy called Zolgensma. This works really well in ours. So here's a 90% reduction again. Here we go, 70% or so in this experiment. And you can see this really nice reduction again here. I mentioned you can make enzymatically inactive forms. We call them M4I. So this goes in, but it doesn't cleave herpes, and that doesn't dent the reactivation at all. So we have a simplified regimen that you can actually think about something like this, probably not M4 based, but something like this, a single AAV with a single meganuclease. You could actually think about getting approved for a human clinical trial. Of course, this is all HSV1. Those are the enzymes we got from Selectis. Lots of folks have HSV2 infections as well, so we'd like to address those. We've had to go out and make our own enzymes for that. Those have been done through a collaboration with Barry Stoddard here at the Hutch, along with Abby. There's only a few groups in the world that really can redesign meganuclease specificity. And so we're really happy to have this collaboration. And the idea, just in a nutshell, is you have a meganuclease wild type. Here's one called Ionu1. We make a version that is thermostable, so it's good for genetic engineering. And it recognizes this sequence, which has a bunch of changes from what we'd like to recognize the closest target sequence in HSV2. Um, and we kind of go through this and start just modifying the enzyme so that it will tolerate this change that it's got that herpes has from the wild type, and then those plus these, and so forth. So this iterative process. At each step, the meganuclease is expressed on the surface of yeast, either in conditions that will not allow cleavage or that will, and we collect all the individual library components that actually will work, enrich those, and move forward. Oops, where am I? Sorry that this is twitchy. And at the end of this, you actually will come out with enzymes that cleave the desired target. Then you can actually just put them into a test tube reaction like this. So here's one called 1831, for example, and it cleaves the HSV sequence really well. You can see controls don't at all. So we have a wide variety of these we're really excited about. Kim in the lab has gone on to show that these actually not only work on the surface of yeast, but they actually work on mammalian cells, and we actually have to tweak them a little bit to ensure stability within mammalian cells. There's a few nuances. Thermal stability is important and so forth. So we now have a series of enzymes that work really nicely. You can see some of the numbers here, and there's even a later generation now that have really good activity, and these are going into animals now to really evaluate our ability to knock out HSV2. Some of these early generation ones for HSV2 we've put into animals. I will say that they're very well tolerated. Obviously, safety for a non-lethal infection like a generally non-lethal infection like HSV is important. We always look at weight gain. The mice in our experiment, the mice are always getting fatter as we have them. But under certain types of AAV therapy, you'll see sort of a loss of weight gain. And that's where we know that they're starting to not tolerate it well. It's actually one of the most sensitive approaches we have is simply weighing the mice. But you can do other things. You can look in the liver for inflammatory cell foci. You can look in ganglia for evidence of neuronal stress that's been described with AAV therapies. We're really not seeing that with these therapies at the therapeutic dose. And in preliminary experiments, these enzymes seem to have a really nice and strong anti-shedding effect against HSV2. This is just like the experiments I showed you. In the control animals, 6 of 10 reacted with HSV2, and depending on which of the clones that we treat them with, it goes from a worst 3 of 9, and then a couple only had a single reactivation event. So it looks like we're on the right track, and we have a later generation that we're even more excited about. I want to mention a little bit, go a little deeper into the safety issue because there's just kind of some cool things that we've done here. So AAV therapy in general, the dose limiting toxicity is liver toxicity. And what starts to happen as the dose goes up is you see inflammatory cell foci. You'll start to get ALT, AST elevations, and this can lead to hepatic failure, ultimately get high enough. And this is dose dependent. So typically our therapeutic dose is 1 times 10 to the 12th. That's well tolerated. If you go up twofold, we start to get into what we call kind of a high dose therapy, 2 times 10 to the 12th, and you start to see more of these inflammatory cell foci. The mice are still okay. At about 3 times 10 to the 12th, it's about as high as we can go. And that's actually consistent across species as well. There are these potential neurotoxic effects. So the neurons after AAV therapy have been reported in multiple species to sometimes show signs of stress. These hypnotic nuclei within the ganglia. This never seems to actually manifest in any symptoms. It has been described, and it's something the field keeps watching out for, and FDA is aware of. AAV's gone into thousands of humans, and we haven't seen any paresthesias or anything like that so far. We high dose therapy. I can show you that the mice kind of stop gaining weight. In fact, these mice maybe are even losing a little bit of weight. So that's if we're expressing a high dose of M4 under the control of an ubiquitous promoter, so lots of liver expression. If we put the transgene under a neuron-specific promoter, the mice tolerate it wonderfully. They don't care. Because there's little, if any, transgene expression in the liver, so they don't get liver inflammation. And we actually don't know what the maximum dose is of this. It's more than we care to put into a mouse just for cost reasons. But the other interesting thing, and not too surprising, you help the liver out, but we actually see less, if any, ganglionic toxicity with those constructs as well. So it really makes the inflammation, the axonopathy, go away as well. So our interpretation, what's going on here, is that the ganglionic toxicity is not a direct effect on neurons. It's actually an effect on supporting cells within the ganglia. That's more of a hypothesis probably than something I'm sure about, but it's completely consistent with our findings to date. The other cool thing about these sorts of approaches is you can start to tackle a general concern in the gene editing field. Now, gene editing, there are approved gene editing therapies for humans, products. There have been approved trials for in vivo gene editing in humans for HIV, for example, and they've been completely safe. But we always worry about off-target effects. So will the nuclease cleave something in the human genome we didn't want? So we've looked a lot at the profile of M4. Again, this is not particularly high optimized. It does cleave HSV. It wasn't designed to not cleave anything else. So it has some off-target sites. So this is just guide tag is a nomination approach where you're actually putting a tag into cells that will integrate into sites of probable cleavage within the genome. If you compare that to a well-known guide for Cas9 targeting a PCKS9 that's involved in cholesterol synthesis in the liver, M4 is below that. I don't want to make a case that meganucleases as a class are better than Cas9 based on this data. Some people will argue that based on other things, but at least these two were certainly not worse. But if you look at these sites, you can detect cleavage of those sites in vivo in our animals. So these off-target effects are real. The animals are healthy as far as we can tell, but these effects have happened. So the sort of worst case scenario, a relatively non-specific nuclease that does what we want, gives us a chance to look at ways that we might minimize off-target effects. So we've put the nuclease again under this neuron-specific promoter, this CAM kinase 2 promoter, or we've used the binding site for a liver-specific microRNA to prevent transgene expression in the liver. So then if we look for off-target effects in the liver under either of those approaches, it's actually reduced 100-fold. So the principle here that I think needs to be applied in general for gene editing therapies is going to be really important to think about what is it that you are trying to edit if you're doing this in vivo, and then really to the maximum extent possible limit the gene editing expression to those tissues. And you can really get around problems of off-target gene editing. Now, later generation meganucleases or Cas9 will have far less than this, 10, even 100-fold less than this just at baseline. And then when you can think about pushing that down another 100-fold, I think you'll end up with a really, really safe therapy. We're getting close to the end. We have a really cool project that I want to introduce in 5 minutes or so, based on an alternative called a gene drive that we've applied to viruses. So you might know gene drives. They're typically thought of in diploid organisms where a gene drive exists on one of the two chromosomes. But actually, let's start with Mendelian genetics first, right? You have some variation, and of course, this gets inherited, but only half of the offspring have it, right? That's basic genetics. Gene drives push themselves into the other genome. Typically, they contain a nuclease that cleaves the other chromosome, and then that's repaired by homologous recombination. They push themselves into this. And so if that happens and this pushes itself into the other chromosome here, when this mosquito mates with another, all the offspring have one copy, which can then push itself into the other chromosome. So these things spread through populations. So they're being used as a way to potentially control malaria spread and mosquitoes, but organisms have to be diploid. The only place it can happen, right? Well, Marius Walter, who was working with Eric Verdin at the time, had a really important thought, which was if viruses co-infect cells, essentially you have a polyploid situation there where gene drive can happen from one infecting virus to another co-infecting virus. And he showed this for CMV, where a gene drive, if there was sort of a 50-50 mix put into acute CMV infection in culture, it would actually drive itself into the other virus. And you could even do this from very, very low starting amounts of the gene drive virus. And it would really penetrate quite well through the infection. So Marius finished up with Eric and wanted to come to the lab and ask what would be a good place to potentially use this therapeutically. HSV seemed good, so would it work for HSV? Could we do it in vivo? Could we do it for a latent infection? So here's sort of the general model, right? In the first embodiment here that we reported last year, it just goes into a neutral locus between this UL 37 and 38. But the gene drive is simply a Cas9 that cleaves its cognate site in the other virus, but it'll bring over, if this serves as a repair template, this will end up in the other virus. So Marius is really clever, and he marks these all things with various fluorophores. The wild-type virus is yellow, the starting gene drive virus is blue, and then the gene drive locus itself is red. So you can actually watch this red locus copy itself into, it starts out in this blue one, but it can now copy itself and end up in the yellow one. And then you can see it because the resulting plaques are orange. In the interest of time, I won't go through this in much detail, but these things grow normally. That's all these are, just growth curves over here, but they do propagate themselves through populations just like you would expect. And these gene drive resulting viruses are the different combinations of starting sources of the gene drive plus it getting pushed into the wild type virus like you would expect. Goes to different parts of the brain with different efficiencies. I think I won't go through that in any detail. But Marius did a very complicated experiment to simply ask the question, can this happen in latent infection? Everything else so far has been acute infection. But what happens if you have a primary infection with a wild-type virus, you establish latency, and then you come in a month later with a gene-drive virus? Can it drive itself into those? And Marius looked at this in two ways. First of all, he just looked at setting this all up and then taking these mice, reactivating the virus, and then doing a clever digital PCR assay on the viruses that were shed and asking are any recombinant where the gene drive itself has been pushed into sort of the yellow virus. And in fact, the answer is yes, you see that. So this recombination between a new virus and a latent virus can happen. It actually happens in both viruses, even if the gene drive aspect of the virus, if there's not the right guide, so it's not actually pushing itself, recombination is still happening de novo. But then if he looks and actually asks what's the rate of that in the viruses with an intact gene drive versus one where there's an irrelevant guide, so there's no actual cleavage, it is happening at a statistically significant higher rate in some animals to a pretty high level. So nowhere near what we've seen in the other stuff where we're 98, 99%. Here we're still in those low percents, but it's early days and it's a completely different approach to targeting herpes. So another shot on goal for our overall goal of trying to help folks living with these viruses. So I'll wrap up. What do we know? Or, what have I told you? That I really think that these herpes viruses are probably the best viruses to actually be thinking about cure studies. I think HIV is incredibly difficult. Hepatitis B is tough. Herpes has a lot of really attractive components. So I wish there were more people working in this field. Gene editing for viruses is a real thing, and it can work really quite well. As I mentioned, we can get up to this 98, 99% after genital disease. And that reduction in viral load leads to a dramatic reduction in viral shedding. I actually didn't show you any symptoms where the mice don't have symptoms. We've done working guinea pigs, and you have this similar reduction in symptoms as well. I should have showed you that. The therapy appears to be safe, especially if you use neuron-specific promoters or microRNA approaches to really limit expression to the places we need it. And then finally, just kind of introduce this idea of viral gene drives. Clearly can happen within viral infections, and can also happen with latent HSV. And so we'll see whether that gets legs as something that they can actually move toward really making a meaningful dent in these infections. And with that, I will stop. This is the group. We're all eating sushi across the street from our area here at the lab working, as well as all the funders. You can read them all here. I always do call out that we've been supported by a large number of individuals through just small and sometimes a little bit bigger donations. And we're always grateful to the people who care about this work. So with that, I'll stop. Thanks for your attention, and I'm happy to answer any questions.
Thanks so much, Keith. This is very impressive, and we are really fortunate to have you sharing with us all this work that you have been doing over the years. I think this is more than 30 years of work. I don't want to date you, but
well, this isn't quite 30 years on this. We did transition in a lab about 12 or 14 years ago, but it's still been a long process.
Yeah. I think, Alan Wayne, please go ahead and ask your question. And I have a couple of more points in the chat that I will just share with you. Alan? Oh, you're muted.
Well, they let me. I had to get permission to unmute. So,
oh, yeah, correct.
You. Congratulations, amazing work, and appreciate the presentation. Great application, relevance to VZV as well. Are you working as hard as it is to make?
We do have some pretty good alternatives for zoster now. I mean, we know Shingrix can be pretty effective at preventing reactivations. Herpes vaccines for simplex have been very difficult, and I remain a bit pessimistic. I hope I'm wrong, and there's some mRNA stuff that we should hear about in the next few months. Hopefully that'll all work. But yeah, it's completely logical, and it could certainly work. And if anybody wants to do it, I will be happy to advise.
Well, there's a ton in the chat, and somebody from Brazil looks like they'll come work with you. Anyway, thank you again.
Yeah, so I'm following up some of these points. So I think, Keith, some questions are like, have you used other animal models to test the herpes gene editing approach?
Yeah, so there's some other mouse. So there's a mouse model, the flank infection model with actual fur plucking. Which is interesting. I didn't realize this, that mice have different hair than we do, and their hair follicles actually release with just if you pull gently, they just come out, unlike ours where it actually hurts and pulls. So it doesn't bother the mice, but it's just enough of a stimulus, oddly enough, that it actually reactivates herpes. So you get the shedding. But it also causes lesions. So our JQ1 doesn't cause lesions. So we can see shedding, but not does it help? And of course, it's another thing we'd like to do. So we're looking there. So that's an interesting model. I don't have anything enough. That's kind of new for us. And I don't have anything enough to say one way or another. But we have done the guinea pig stuff I alluded to really quickly. The guinea pigs are a lot of work. I originally underestimated, I think, how much more difficult they are to handle than mice. They're about 10 times realistically, just in kind of throughput. But they have spontaneous reactivation. You don't have to give them anything or pluck anything, so that the virus reactivates. And they get lesions that you can see and score, and they look a lot like a human lesion. So there we're seeing similar effects. So we're seeing effects on shedding. We're seeing effects on the symptomatic disease as well. So those results we haven't actually published any of that yet. We just want to have a big enough body of work so that it's something meaningful besides just kind of one experiment.
And do you think, and this is another point, do you see this moving into human clinical trials anytime soon?
Yeah, for sure. I mean, that's our goal. Absolutely. And one nice thing I mentioned, we simplified this down to an AAV9 based regimen. AAV9's gone into tons of people. So AAV9 is the basic. It's the AV type Zolgensma, which is used for spinobulbar muscular atrophy. It's an approved product. AV9 was also the the AAV in the Excision Biosciences HIV-1 trial. So that was a AAV9 delivering Cas9 targeting HIV at similar doses to what we would do, completely tolerated, no adverse effects. Obviously, they weren't really trying to target the ganglia, but we know that AAV9 goes to the ganglia in humans. Um, so yeah, fortunately, this is a field that's moving forward sort of on different fronts, but they're all helpful to each other. There's not a ton of work on herpes simplex, unfortunately. There's a little bit out
Mm.
there and getting to be more. But we can benefit from folks working on HIV or just inborn errors of metabolism and so forth, and really lean on those studies for safety and understanding how to make this a real therapy.
So realistically, and so many people are actually enthusiastic to know, like, what is the timeline looking like for
Yeah,
clinical
well,
trials?
I can't say on the timeline other than we're working hard on that. There are optimization and a set of safety studies that just need to be
Mm.
done to get approval even for a trial. And there are manufacturing issues and how do you actually make enough of this? And we are working through all those, but those aren't really scientific things sometimes where you can just
Yeah,
show a slide in the
no,
data, but they're
absolutely.
happening for sure.
And we would love to help you manufacture these products in patients.
I'll bet. Yeah.
Um, yeah. And one question is asking, how do you determine the viral load, the latent viral load? So is it by the number of viral particles introduced upon infection, or does it change with the reactivation?
Yeah, oh, that's a great question. So we collect the ganglia and then we basically have a digital PCR assay we use for this because digital PCR is so precise that you can look at a 50% load reduction and be confident in it. And then everything's normalized. So we do that at the same time relative to just total viral input. So it's usually expressed in viral load per million cells is how we express that. Um, so that's simply collecting that we have looked. That was a really important question. Does the reactivation process change the viral load? And the answer is no. That's actually in one of our papers. We reactivated the mice 7 times over a multi-month period. And it's one of the most beautiful pieces of data that we've ever generated because it almost looks too good to be real because digital PCR is so precise. There's a clustering in the animal groups is perfect. Of course, and you have to sack the group to get that. So we started with however 80 mice and carried them forward. And there's absolutely no change. I mean, there's these tight clusters that just don't move up or down whatsoever. So I think the ganglionic load really seems to be set, and it doesn't really seem to replenish itself as far as we can tell
Mm.
through these. So once we get it down and then reactivate, it doesn't seem to come back up. So I think that really bodes well for the durability of a therapy. If, for example, we were in a human and we got rid of 99%, and so now there's not clinical reactivations anymore. It's not like it's going to get worse later on. I mean, that's what our data is suggesting, but obviously that'll all need to be tested.
Mm. No, that's interesting. And another point also here is from the same person who asked this question is, do you think there's a concern with the recombinant herpes simplex viruses to be less conductive to cutting or editing? Um,
So we and others have certainly observed resistance to cutting in viruses that turn themselves over and mutate a lot. So
mm,
HIV, that can happen, hepatitis B, that can happen. Um, just because the fidelity of replication of those viruses is low, and so
right.
if anything, herpes is not turning itself over in the neurons, and there's very little genetic diversity in herpes. So one of the things, if you're designing these sorts of therapies, as we are for hepatitis B, and we've worked in HIV as well, you really have to look at conserved regions of those viruses and regions that can't really change because you want to be able to hit all this wide variety of viruses. It's not really a problem for HIV. The genetic diversity is tiny compared to those, and then it doesn't drift over time. So we've never seen any evidence of that whatsoever in
Mm,
HSV. Um,
mm.
so no, I'm actually not worried about that here.
Okay. And do you plan to advance this approach for HSV1 or only for HSV2?
We're working on both actively, for sure. Yeah. Yeah, and those same approaches for where we're kind of making de novo the HSV2 and meganucleases, we're also trying to develop a much better set of HSV1 enzymes as well, because these original ones just really were never intended for ultimate human use.
Right.
They do have these off-target effects. But I kind of like them personally as sort of this worst case scenario. Like these actually have a lot of off-target activity compared to like new generation enzymes. And you know this for Cas9, for example, now these high fidelity approaches that have much less off-target effect than the original enzymes do. And those are just going to be safer. You want this to be as safe as possible. And so even in these worst case scenarios, the animals tolerate it just fine. So as we do these tricks, I think we're going to have something that we can put into human beings with confidence.
And when you saw clearance in mice up to what, 98, 99%,
Right.
do you think this 1-2% might replicate again and cause an outbreak?
Um, well, certainly we still do have some reactivations in those treated mice. Most of the reactivation studies we've done are in the orofacial model. And remember that although, I like to, it's 98, 99% in the genital tract, after orofacial, it's 90% for the SCG and 60-ish% for the TG. So we still see this dramatic effect even there. I mean, most people really thought that the TG was the main part of reactivation. I'm not sure that that's really true. And I think people who really thought herpes a lot know that the autonomic ganglia, like the SCG, are super important. So we're not seeing this kind of reseeding thing. And the other point I'll make is that just for reasons of practicality, we evaluate the mice after 1 month of therapy, but these are episomal delivery vectors in non-dividing neurons, which means that there's still expression there at the end of a month. And we've done this analysis that gene editing is still continuing over time. So it's actually getting better. So it's not like it stopped in a month or that's the end point. So we just really need to do the experiments, but they're to take a lot of resources. Is at the end of 6 months, I'm not quite sure the number where we'll be, but it's going to be better than that. What is it, a log better? I don't really know yet.
One last question on this, and then I have a question on the gene drive, which actually I found it very fascinating. So one last question also is relates to the viral load that you have been observing among different animals.